So, very good morning, students. Today we are going to discuss the entomopathogenic nematode in the course of the invertebrate pathology. As you all know, the entomopathogenic nematodes are the soft-bodied, non-segmented roundworms, so that are obligate and sometimes facultative parasites of the insects. Obligate parasites mean so they need a living host, otherwise they will not survive. And sometimes facultative parasites mean so in the insects they can live in the other insects, but under the suitable cost available only, so they can act as a parasites. So that is what we call it as an entomopathogenic nematode, mean their target is to attack insects. So entomopathogenic nematodes are soil inhabiting. So normally you can expect them in the soil where these insect, non-motile insects or insect stages are there in the soil, they can survive associated with these insects. The lethal insect parasites, so definitely it can kill. Right? Once the insect attacked by the nematodes, the insect will be done. So that belongs to phylum Nematoda. You all know the phylum Nematoda. There are plant pathogenic nematodes are there, entomopathogenic nematodes are there. But among the phylum Nematoda, so there are two families. I have took some of these section in the nematology class 2 in the third year second semester. The Steinomatidae and Heterorhabdidae. There are two families are very important. So species from these two families, the genus Heterorhabda and Xenorhabda, two genus. From these two genus, there are several species available, commercially available. They are very important, host specific. They have proven to be most effective biological control organism of soil and above ground pest. So in comparison, if you are comparing with the other entomopathogenic fungi and bacteria, protozoa, and viruses, these nematode also to be proved as effective biological control agents. So if you are taking that the entomopathogenic nematode history, so first, so the 17th century they have identified, they have isolated, but they did not know much about information about these entomopathogenic nematodes. But to control the pest started in 1930s, right? 1930s. So the entomopathogenic nematodes are very fit with the integrated pest management or IPM programs. So because they are considered non-toxic to the human and relatively specific to the target pest. So therefore the non-target effect is very minimal in comparison to the entomopathogenic bacteria and fungi. Right? It can be applied with the standard pesticide equipment. You can mix with the even chemicals, you can spray. Right? So if you are taking the phylogenetic position of these entomopathogenic nematodes, these two families, so belongs to kingdom Animalia and phylum Nematoda. So we call it as roundworms. The eelworms belongs to phylum Nematoda and class Chromadoriae. Right? In the class Chromadoriae, order Rhabdida. In the order Rhabdida, there are two families, important families. One is Steinomatidae, other one is Heterorhabdidae. These two families, from the Steinomatidae, Steinonema genus, and Heterorhabdidae, Heterorhabditis, these two genus are very, very important. From these two genus, there are several species. Steinomatidae, we will study in the coming slides. Steinomatidae, Carpocapsi, Heterorhabditis, Rhodopola. So there are several species are available. So these species are nowadays commercially available. The asset for mutation, so ready-made you can use it. So if you are taking the history of entomopathogenic nematode, it's a brief history. EPNs was first collected by Krusi, a scientist in Germany, from the Hymenoptera. Right, so Hymenoptera sawflies, sawflies pupa, he isolated. So in 1970, right, now in the 1970s, they are exempt from the registration and regulation of regulation requirement by the Environmental Protection Agency of the USA and the Central Insecticide Board and Registration Committee of India and other similar agencies of many other countries, because they did not know much information regarding the target effects. So they thought initially that so nematodes can cause several nematodes can cause infection to human being, no? But they did not know much information regarding these entomopathogenic nematode. So therefore, they are reluctant to register this one as a biocontrol agent. Thereafter, the CSIR, so in Canberra, it is located in the Canberra Commonwealth Scientific and Industrial Research Organization. It's a leading research organization in Australia, first commercialized. So it's registered by the Australian Environmental Protection Agency and registered, then produced commercially, and to manage the wine beetles, the vines. So the grapes wines are very big, the bean vines are very big problems because they will cut it. They use this EPN. So because the environmental conditions are very suitable and the grapes flowering and fruiting seasons, very due condition. So nematode will work. Its current and the current borer moth, these two organisms, against these two organisms they used. So if you are taking the global distribution of this is an old map, but nowadays in other countries also it has been identified. So Steiner nematode is indicated by the red star, and the Heterorhabdidae indicated by the blue spots. So where you can find now where these nematodes, the entomopathogenic nematodes, has been identified. Even some other countries, they haven't took much researches on that. The two genera, Steinenema and Heterorhabditis, right? So among these two genera, for more than 64 species in the Steinenema and 8 species in Heterorhabditis and 1 species of Neostinema. So this Neostinema is the very old entomopathogenic nematode species, or even nowadays they are using. So before the Heterorhabditis Steinenema, Neostinema they is identified earlier, right? So these are the pictures showing that how if insect cadaver, so we call it as a cadaver means dead insect, or if you are taking the non-motile insect, how you can see this entomopathogenic nematode will be there. So the root weevil infected by the EPN, you see inside the grub body is filled with the nematodes. So thousands and millions of nematodes. Here, this pupa. Nematode are existing the waxmoth pupa. You see from the waxmoth pupa how many nematodes, so millions are coming out. So in this way, so these entomopathogenic nematodes are very well suited biocontrol, very effective biocontrol agents because of these habits they have, right? High proliferation. We will study in forthcoming slides how these life cycles, right? So you all know this entomopathogenic nematode in the life cycle. If you are taking the life cycle of these entomopathogenic nematodes, if you are taking from the egg, right? If you are taking from the eggs, so juveniles will come out, right? Juveniles, first stage juveniles is an only free living stage of entomopathogenic nematodes, right? So this infective juvenile stage is the only free living stage of the entomopathogenic nematodes, means J1. So the J2 penetrates, juvenile penetrate the host insects through the natural openings or where the soft tissues are present in this insect body, right? So if you're taking the natural openings, spiracles and the mouth and anus, these are the natural openings. Other than that, you can see that very soft membranes in between the intersegmental membranes, right, in between the sclerites. So it's sclerites connected with the soft membrane, so they can easily penetrate these soft membranes and it can go in and attack. Then enter into the hemocoel. Finally, if it is entered into the natural openings, so these soft cuticles, then finally it will reach the hemocoel, right? So both Heterorhabditis and Steinonema are mutualistically associated with the bacterial genera. There are two bacterial genera. Photorhabdus associated with the Heterorhabditis, Xenorhabdus associated with the Steinonema. Right? These mutually associated with the bacteria always will be there in these nematode guts. Right? So once the nematodes reach the hemocoel, right? Once a nematode reach the hemocoel, this bacteria will work. Right? Bacteria will work. So if you are taking that this life cycle of these nematodes, right? For this first generation, so the death of these insects, from the death insect, the first generation hermaphrodite. There are some parthenogenetically they can multiply, and hermaphrodite will be also there. Right? This first generation hermaphrodite, the second amphimictic male and female. Right? Second generation amphimictic male and female. Amphimictic male and female means so sexual differentiation will be there. Right? So hermaphrodite means in one, for a single nematode can have these both reproductive. That's what we call it as a hermaphrodite. Here amphimictic male and female will be there, so they will copulate, and from this emergence, then third stage infected juveniles. Right? Third stage penetration of the insect, insert the bacteria and kill them. So death can occur in 24 to 48 hours. This is tells us how these nematodes are very fast to kill these host, right? Kill these host. So juvenile stage release cells of these symbiotic bacteria. I told you there are Photorhabdus and Xenorhabdus bacteria associated with these nematodes. Once this nematode reaches the hemocoel, they will release the symbiotic bacteria from the intestine into the hemocoel. So what will happen if the bacteria reach at the hemocoel? We will call it as septicemia. It's a bacterial infection of the insect blood. So any blood of any organism, we call it as an infected with this bacteria, we call it as an septicemia. Mean in blood get infected, mean finally will be death. As septic. Yeah? So bacteria multiply in the insect hemolymph, and it's infected host usually dies within 24 to 48 hours because of this septicemia. Right? So because these bacteria produce toxin, this toxin react with an antibody of these host cells. So then finally it will break the antibody, and finally it will finish these insects. Insect will die. After death of the host, nematodes continue to feed the host tissue and mature and reproduce. You see, after the death of this insect, the nematode working. So how this association works, right? How this association works in the coming slide will explain how this bacteria and nematode associated, how both each and everything, the bacteria and nematode help each other. And progeny nematodes develop through the 4 juvenile stages to the adult. J1, J2, J3, and J4. And again, it will mature, sexually mature and die. Egg laying eggs. In this way, this life cycle of this nematode completes. Depending on the available resources, one or more generation may occur within a host cadaver. A number of infected juveniles are eventually released into the environment to infect other hosts and continue their life cycles. You see, if the infected host is having abundance of food, or more than pupa, more than one pupa, or more than one host is in that area, it means abundance of food. So therefore, they can able to complete its life cycle 2 to 3 times. Right? 2 to 3 times. So reproduction differ in hermaphrodite. So heterorhabdic and xenorhabdic nematodes. Reproduction differ. It's that's what I told you in the life cycle. Ambiguous males and females will be there. And here the hermaphrodite males will be hermaphrodite reproductive stages will be there, right? So the infected juveniles of the heterorhabdic nematode become a hermaphrodite, right? The heterorhabditis always reproduce by hermaphroditic adults. Individual of the next generation produce both males and females, wherein stenonematic nematodes, whole generation are produced by the males and females, call it as a gonochorism. Gonochorism in MVP. It's gonochorism. So insect cadaver become red if insects are killed by the Heterorhabditis. If the Heterorhabditis is attacked, insect cadaver, dead insect will become a red. So if you are finding any red insect pupa, then you have to check it whether it is infected with nematodes. There will be chance of this rainy season, right? And brown and tan if it is killed by the strain. Brownish color is showing that brown or tan brown, not dark brown. Dark brown is as usual healthy pupa, right? Healthy pupa. And if it is a brownish lava, then you can suspect, right? Tan if it is killed by the strain. So you have to be very careful on this. Heterorhabditis attack that is in reddish color. Stenonemitis is attacked in brownish color. Heterorhabditis is always reproduced with the hermaphrodite. Stenonemitis is always reproduced by this gonochorism for the amphibians. Nice. So color of the host body is inductive of the pigment produced by the monochromatic bacteria growing in the host. Right? So this color changes because of the pigment produced by the bacteria. So if you are then this is an indication of that when you are going to culture this bacteria artificially, you can culture it. Right? Nowadays technologies are available. You can culture it artificially. So if you are artificially culturing, so again you need a nematode. Otherwise it will not go into that insect now. So artificially culture it, then it finally. So what you can do is you can mix up with, and I will later tell you the technology how it is worked. The color of the body because of this pigment produced by the infected bacteria. It's monoculture, right? Monoculture bacteria. This is the thing. So I have to explain that one. The Steinonema associated with the Xenoraptors, the summary. Heterorhabdida associated with the Photoraptors, right? Hetero, photo. Steinonema, Xenoraptors, right? So J3 is an infective stages. J2 to I told you J2 and J2 to J3, right? J3 infective stages. Male and female first generation, male and female second generation, and 8 to 10 days the J2 will complete the again will become an infect in 8 to 10 days. Generation can complete in 8 to 10 days. It's J3, male and female, and again your life cycle. If you're taking the Heterorhabditis, first generation is hermaphrodite. Second generation, you can expect male and female. It completes a life cycle in 12 days. Right? It completes in life cycle in 12 days. That's what the Heterorhabditis hermaphrodite. This is a gonochorism or amphibians. So you can now study the searching behavior of the nematodes. I already explained that in the last semester too. Entomopathogenic nematodes uses two searching strategies. One is we call it as an ambitious, other one is we call it as an crucious. There are two strategies, ambitious or crucious. Ambitious normally used by the Steinonema carpocapsy. For example, if you are taking the Steinonema carpocapsy uses ambitious, have an energy conserving approach and lying wait to attack mobile insect in the upper soil. Right? We call it as a mobile insect, we call it as a nictitating. Right? Nictitating means, so if the insect is mobile, we call it as a nictitating. So to serve the energy, what they will do is, it will be there as it is. If the insect is mobile, insect is moving, suddenly it will attack. Right? That is we call as an ambush. Claymore attacks. Nowadays, the word people are using, right? Claymore attack, because of the vehicle is moving vehicle to attack. Use claymores because it's an ambush. That's what we call it. Cruises, like the Stenema glacier. Right? Stenema glacieri and Heterorhabditis bacteriophora, these two species, normally they have identified, they are using the crucious techniques. So the crucious insects are very efficient compared to the ambitious. Why? So crucious, we can target for the pupils, non-mobile insects. Ambitious we can target for the larva. Normally the larva will be there above ground soil. So therefore sometimes if the environmental condition is not suitable, it will not work. But if it is working under the soil, so we can give the moisture it, and if you are maintaining the moisture and if you are inoculating this Cyanema, Glaciaria and Heterodapletis bacteriophorae, definitely they can kill the pupal stages. Highly active and generally subterranean under the soil, moving significant distances using the volatile cues. Where the host is there, from these cues, chemical substances emitting from that host, it will move it. Right? It will move it. Other methods to find the host, the underground. Cues and other methods. So therefore, they are effective against less mobile pests such as white grubs. Scarabid beetles, white grubs, always grubs will be there in the soil. Some nematode species such as Steinanima felti, Steinanima feltiae and Steinanima riobrave. These two other species, Steinanima feltiae, Steinanima riobrave. These uses intermediate foraging strategy. Intermediate foraging strategy, combination of ambush and crucius. These some other nematodes, these two, Steinanima feltiae, Steinanima riobrave, these two species are using the both techniques. Right? They can use the ambush as well as a crucial techniques. So this is the indication of this bacteria. This is this electron microscopic picture. It's showing that how this fluorescent, this is fluorescent dye, right? Dye given the color of this the bacteria, right? Color of the bacteria, the Photorhabdus bacteria, how it is associated with the gut of these entomopathogenic nematodes, Photorhabdus, greenish color. You see that they are changing that body cavity, especially the guts, into the bacterial chamber for the survival. As like you would have studied in the rhizobium. Rhizobium changing or modification of the root hairs to become a knots, right? The modification and knots, then it changing the cavity now. So that's the way it is working. That's the way it's working. So it's a color, body is changing. So here this picture also clearly showing that nematode have association with the bacteria. Right? Nematode with the association with the bacteria. Wait for a, we will continue, right? So this anterior part of the intestine of the infective juvenile is modified as a bacterial chamber, right? So right, like the rhizobium, how it is modified in the root galls, like that this, if the intestine is modifying where this chamber, in this chamber, you can see this bacteria. That's the thing we call it as a close association. This association we call it as a symbiotic relations. So how this nematode and bacterium complex works, right? So for this nematode depend on the bacterium, bacteria depend on the nematode. For what purpose this nematode depend on the bacteria? First thing, kill its host. Nematode cannot kill. Nematode can only penetrate and go into the hemosyl. To kill its host, it needs bacteria. Right? Bacterial toxin can kill the host, creating a suitable environment for nematode development and prevent the secondary infection by the microbes. So this bacteria helps to development of the secondary microbes because it producing toxins. Secondary microbes will not grow. And also gaining suitable environment for the nematode development. If the antibody present, if the host antibody will present, then nematode will not survive. So therefore, need to create a suitable environment. Bacterium can help to create a suitable environment inside the insect body and serving as a food source. Right? Bacteria also is serving as a food source. But for the nematode also consume these bacteria. Break down the host tissues to serve as a nutrient source. Right? Bacteria will produce certain enzymes. These enzymes will break down these host food then the host food sources will be eaten away by this. For these four reasons, nematodes depends on bacterium. So bacterium also depends on nematodes to protect the external environment. So bacterium cannot survive without the conditions like this chamber present in the nematode, right? So therefore, in the outside environment, it will not survive. Once it's reached the inside hemocoel, they can survive. So therefore, it needs protection from the external environment. That's how the nematode giving an protection. Both, right? So bacterium giving protection from the insect environment, nematode will giving the protection from the external environment to the bacteria. You have to be very careful, and you should clearly know that. Right? Penetration into the host hemocoel. For the host hemocoel penetration, it needs a weapon. These weapons are nematodes, right? Like a missiles, right? So bomb will be there in the missile. Missile is propelled. Once it's reached the target, bomb will blast. Like that, this nematode act as a proper vehicle to carry that one. Once it's reached the target, it can. Inhibition of the host antibacterial proteins. Okay? Some antibacterial proteins will be there. This also will be broken down by the nematode digestive enzymes. Right? Nematode digestive enzyme. In this way, this both nematode and the bacterium help each other, and they are survival. Right? They are survival. Nematode arthropod association can be divided into four categories. This association we can categorize into four. Right? One is phoretic. We call it as a phoretic, mean nematodes are transported by an insect. That is we call it as a phoretic. Another one is there necromanic. Necromanic means obtain nutrition from insect cadaver. These are the associations, right? These are the associations. You should remember the terminologies, right? Phoretic means nematode are transmitted by the insect. Entomophilic, hemophilic, like that. Phoretic means nematode transmitted by the. Necromanic is the obtaining nutrition from the insect cadaver. It's called necromanic. Facultative parasitism and obligative parasitism, you will know, you are very clear. Facultative parasitism means if these other hosts are so not suitable hosts are available, so they can survive in the in the other hosts. Obligate means it need the host, otherwise it will not survive. Live host. That's we call obligates. These are terminal. Nematode also interact with bacteria at the at least three ways. One is tropism. Nematode eat bacteria. Parasitism, pathogen cause nematode diseases, and mutualism, nematode and bacteria. Okay. These are the terminologies you should be very careful. Now, why these nematode and arthropod associations are there, and how the nematode and bacterial associations are there. So, so if you won't ask any question, so this is a so far we have discussed the what are the categories of entomopathogenic nematode, where they are living habits, and is how these bacteria associated with this nematode, what are the mode of action of these entomopathogenic nematode, how these life cycles completed in different nematodes in different ways, and why these bacteria needed for the nematode, why these bacteria nematode needed for bacteria. So those are the things we have discussed, and some technological term for these interferences, association of these arthropod nematode bacterium interactions. So now we are going to discuss the isolation of these symbiotic bacteria. So as I told you earlier, so you can, if you want to produce commercially the entomopathogenic nematode, you can go for 2-3 days. One is you can get the nematode. From this nematode, you can isolate the bacteria, and you can mass multiply this bacteria, and you can use with the other technology to spray without nematode. Other way is there, you have to isolate the nematodes, you have to mass multiply the nematode, you have to give artificial food and other things, you can make a commercial formulation of the nematode, you can use it. Right? So these two ways, we nowadays the biopesticide industries are developing the biopesticides. The first thing is we have to know the how symbiotic bacteria can be isolated, how you can going to culture it. First, the symbiotic bacteria you can isolate from the cadaver, because if the cadaver is showing the reddish color or the cadaver going the thin brown color, so what you can do is you can collect that cadaver, surface sterilize that cadaver, then from that cadaver you can take out the inner content from the larva and you can streak in a suitable medium. This suitable medium is we call it as a Macongi agar plates. You can Macongi medium, you can use it. In the Macongi medium, if it is giving the red color, it's an indication that okay, this symbiotic bacteria we have isolated from this cadaver. So once this Macongi agar, using this Macongi agar, you can mass multiply it. These bacteria you can spray with these other chemicals, pesticides, or you can spray directly to the insect. So the insect should eat it, it should reach the hemocyte. That the problem is, so nowadays is the problem is when if you are applying this symbiotic bacteria, whether it will reach the hemocyte or not. It will work if it is reach the hemocyte. Otherwise, it won't work. Right? Like the COVID-19, right? If you are eating the COVID-19, it will not cause a problem. If it is reached the nasal cavity, the lungs, only the problem. Okay? That's how, where it is working, you have to direct that one to reach over there. Right? So primary colony is from the pink color. From this one, you can use it. So this is an isolation of the symbiotic bacteria from the cadaver and mass multiply, and you can formulate the biobactericides. Second one is an extraction of this entomopathogenic nematode. You have to get the soil sample. From the soil sample, you have to isolate by using the baiting method. Right? So you have to collect the soil sample from the root zone, so the way you want to collect it, and bait the Galleria melonilla larva. So Galleria is a waxmoth larva because we are using the waxmoth larva because abundantly available. It is very active. It can move here and there through the soil. While moving, it can attracted by this nematode, and one is infected with this Galleria melonilla, so it can multiply. Many nematode can use this Galleria melonilla as a host, suitable host. That's what we are using. On the collection of the infected larva, once it infected, you collect it. You have to rear it. From this extraction of these infection EPNs from the infected larva, then re-isolate infection and isolation of the EPNs and store it at 8 degrees C. Otherwise, it cannot be storable, it will die it. You have to give artificial diets for further formulations. Okay? For the formulations. So this extraction procedure consisting 2 branches. One is the rearing of waxmoth larva. Other one is rearing and isolation of these entomopathogenic nematode. These two parallelly doing only, you can small scale or commercial scale cultivate production of the entomopathogenic nematode you can do. Otherwise, it is not possible. Okay. So the isolation of these from the entomopathogenic nematode from the host also. First, this is the first culture you have to isolate from the host. But cadaver and soil. Infected host insect larva you can cultivate, or you can get the soil sample and you can bait. You can do the baiting. Waxmoth larva, so incubate it 25 degrees C. If you collected the cadaver, you have to incubate it 25 degrees Celsius for 3 days. Or if you are using the soil baiting also, once it's infected, you have to give a 25 degrees C for mass multiplication of the nematode for 2 days. It will complete the life cycle in 24 to 48 days. But if you are giving 25 degrees C, your multiplication rate will be very high. Right? So then you can examine the larva on 8 to 10 degrees Celsius, you can keep it, right, for the commercial use. The steps involved in the mass production. So we will discuss this mass production in the forthcoming class. And if you won't ask any, I am going to stop here. If you won't ask any question.